Good morning, viewer. Welcome to our Apt English viewers. Scientists analyzing ancient DNA from a 12,000 years old double burial in southern Italy uncovered genetic evidence of a rare inherited growth disorder in two closely related prehistoric individuals. A team led by researchers at the University of Vienna and Leg University Hospital Center has traced a rare inherited growth disorder to two people who lived more than 12,000 years ago. By pairing ancient DNA results with tools used in modern clinical genetics, they identified the condition in a mother and daughter buried together in southern Italy. Reported in a New England Journal of Medicine, the work highlights how paleogenomics is becoming a way to study both ancient population history and the biological roots of disease in individual prehistoric lives. Viewers, the findings comes from a new look at a famous upper Paleolithic burial uncovered in 1963 at Grotta del Romito in southern Italy. The site has remained a scientific mystery because the bones show unusual growth patterns and the burial itself is striking, leaving open questions about who these people were and what might explain their short stature. In case like this, skeletal clues can suggest a disorder, but many conditions can produce similar bone changes, which makes definitive answers hard to reach without genetic evidence. The two individuals were laid to rest together in what appears to be an embrace, that is, Romito 2, an adolescent with a pronounced limb shortening who was long assumed to be male, was found cradled in the arms of Romito I, believed to be an adult female. Researchers reported no signs of trauma. Estimated from skeletal measurement, Romito 2 stood at about 110 centimeters, a height consistent with a rare disorder called acromesomelic dysplasia. Though bone evidence alone could not confirm it, but Romito 1 was also shorter than a typical for the time at about 145 centimeters. For decades, these details fueled a debate over their sex, their relationships, and whether a single underlying cause could link their stature. The team analyzed ancient DNA extracted from the petrous foot of the temporal bone of both individuals, a region known for preserving genetic material well. Viewers, genetic analysis established a first degree relationship. The researchers then screened genes associated with skeletal growth and compared the identified variants with modern clinical data. This interdisciplinary approach, combining paleogenomics, clinical genetics, and physical anthropology, involved an international team from the University of Vienna and collaborators in Italy, Portugal, and Belgium. The analysis showed that both individuals were a female and first degree relatives, most likely a mother and daughter. In Romito 2, researchers identified a homozygous variant in the NPR2 gene, which is essential for bone growth. So, this confirmed a diagnosis of acromesomelic dysplasia Marotex type, and a very rare inherited disorder characterized by severe short stature and marked shortening of the limbs. Genetic data from Romito 1 indicate that she carried one altered copy of the same gene, a condition associated with milder short stature. Where the sex closely report.